Our next speaker is Taia Wang. She's an associate professor at the ITI and the Department of Microbiology and Immunology. She did her MD PhD training at Mount Sinai and then a postdoc with Jeffrey Ravech at the Rockefeller University. She has a strong interest in glycosylation of antibodies and the impact on the adaptive and innate immune system. And I think we hear something about her research in dengue. Thank you, Taia.
Thank you, Toby. Let me get the pointer. Great. So I'd like to start with a little intro on antibody glycosylation because it's a topic that many people are not very familiar with. IgG antibodies, as you know, have two functional domains. Effector function mediated by IgGs are dependent on the structure of the FC domain, which has two main structural components, the protein backbone, otherwise known as the subclass of the IgG, and then the post-translational modifications represented here in blue are the second important structural element. I became interested in glycosylation of human antibodies during my postdoc with Jeff Ravech. At that time, it was known for 20 years that you could change up the sugars on a therapeutic monoclonal antibody and that would affect its activity. But it was not known whether sugars on endogenously produced IgGs are an important regulator of human immunity. And so I'll show you a bit about where this field has gone in the last 10 years and then bring you up to speed on this odyssey that we've been on in trying to understand the role of antibody glycosylation in dengue virus immunity. So endogenously produced human antibodies have two N-linked glycans on them, one on each CH2 domain. Every single CH2 has this core glycan, and then these other sugars are variably present. I'm going to tell you about two sugars. Only two sugars can change an antibody, two antibodies identical in protein sequence with or without these two sugars have completely opposite activities in terms of regulating the inflammatory response of an immune complex. The sugars are this terminal sialic acid and what's called the core fucose. What we know is that in terms of human antibodies, it's useful to bin them functionally into three categories. So the majority of antibodies, about 80% of your IgGs, are modified by this sugar here, which we'll call the neutral or the wild type sugar. It has the core fucose, lacks the sialic acid. There are then two minor populations. One has both of those sugars. So that one I'll refer to as the sialylated form because the sialic acid is the functional determinant of that sugar. The other bin is one that we call the afucosylated form. It actually lacks both sugars, but it's the absence of the fucose that confers a very pro-inflammatory phenotype to that sugar. At a population level, these are inversely regulated so that these are, each dot represents an individual donor. On the y-axis is the abundance of the sialic acid and the sialylated form, and on the x-axis is the afucosylated form. So these are the two minor forms here. What you see is some people are very enriched for the sialylated form here in blue, other people for the afucosylated form, but almost no one is in this upper right-hand quadrant, right? So we have shown that people in this red quadrant are at very high risk for more severe disease during COVID-19 and dengue infection, whereas people in blue here are protected from lung inflammation during influenza virus infection. What regulates these modifications is a very important topic of active discovery. The main correlate that we know of is age. So that with increasing age, you essentially move from this phenotype, which is an anti-inflammatory form, over to the right to the more pro-inflammatory glycoform. Functionally, so how do these two sugars regulate immune function? Represented here in the red triangle, I'm showing the afucosylated immune complex here. So antibodies bound to in vaccine antigen or an infecting viral particle. The afucosylated glycoform has a very specific activity. It increases affinity for CD16, which is an activating Fc gamma receptor, signals through ITAM domains. downstream activating many different functions. Very importantly, secretion of soluble factors that promote inflammation, recruitment of effector cells to the site of immune complex formation. I won't go through all of these functions, but and I would say that this list is still growing. We're still discovering all of the effector functions that can be mediated by these different receptors. These here, what I'm showing are what we call the type 1 Fc gamma receptors. These are the canonical receptors initially discovered about 30 years ago. The sialylated antibody here, represented in the blue triangle, has very different activity. So one is that it shifts binding more toward the inhibitory receptor. CD32B signals through an ITIM domain, and that receptor has the activity of counterbalancing the activating receptors. But the sialylated Fc can also do something entirely different, which is that it shifts specificity of the antibody for what we call the type 2 receptors. So these are C-type lectin receptors, more recently defined as IgG receptors. CD23 is a receptor on B cells that, when triggered by sialylated antibody, and I'll go into this in a little more detail, results in selection of higher affinity B cells during vaccination. CD209 is a receptor that, when engaged by the sialylated Fc, has very potent anti-inflammatory activity. Not just it can block a subsequent response to, say, LPS. And how does this happen? So we know from work by Jeff Ravitch's lab that one consequence of engaging CD209 is upregulation of the inhibitory receptor as a mechanism for increasing the threshold of activation of innate effector cells. We have recently defined a separate mechanism, which is induction of a transcription factor called REST. REST has the activity of blocking transcription of NF-kappa B factors. So ultimately, a separate mechanism for blunting the inflammatory response triggered by immune complexes, but actually we believe sort of any activator of NF-kappa B, and including LPS, as I mentioned, TNF. Okay. So this is just another way of showing you there's a large degree of heterogeneity in the human population of the afucosylated glycoform, and it's quite stable over time. So this is two separate populations. This is a pediatric population on the right, on the left here, in collaboration with Alan Rothman. You see over the course of years, people have very stable levels of these glycoforms. Here, this is a collaboration with Jeff Taubenberger looking pre and post influenza virus infection in a controlled human challenge model. You see almost no change in influenza infection. So very stable over time. Sialylated antibodies, same, very heterogeneous among humans and very stable over time. So really, building the hypothesis that your basal profile of these sugars might affect the immune response. An initial experiment that we did to try and answer whether post-translational modifications of IgGs are important was to vaccinate humans, healthy adults, and just ask, is there any clear regulation of sugars on antibody molecules during an active immune response? That would suggest, if so, that they are important in shaping immunity to some degree. And here's what we saw, is very striking regulation, just looking again at those two sugars. In blue, a boost, this is after influenza virus infection, either pre-baseline or day 7 and subsequent time points. We saw this very clear boost in sialylated antibody production after vaccination. So this, again, is a type 2 receptor binding Fc domain. And interestingly, very clear inverse regulation with that pro-inflammatory form, the afucosylated form. So clear regulation, this suggested a role functionally in maturation of the vaccine response. And this is actually not work I'm going to get into. I'm just going to sort of show you where the data point in terms of how that sialylated antibody in particular is functioning during vaccination. So the model is that the vaccine antigen is engaged by IgGs in circulation. People with higher levels of sialylated antibody in circulation that binds to the vaccine antigen ultimately produce a higher affinity and more broad antibody response after influenza vaccination. We believe that this is the mechanism. So sialylated immune complexes containing sialylated antibody, the sialic acid here represented by the red, are fixed on FDCs in the germinal center. And then B cells, which express CD23, can be engaged by the sialylated antibody in that immune complex. Triggering CD23 on the B cell results in increased expression of Fc gamma R2b, which is that inhibitory receptor. That functions to increase the threshold of activation required for the cell, ultimately resulting in selection of cells producing higher affinity antibody. So this was some first work really demonstrating that antibody glycoforms are actively regulated during immune responses and that they're important in determining the quality of an ongoing immune response. But is there a role for the basal profile of glycosylation on your antibodies in determining outcome during an infection? So that was a study that we initiated some time ago, asking this question: is there a specific type of antibody that's more abundant in people who develop severe dengue disease? And we focused on dengue because antibodies have a clear role in modulating dengue immunity. They can both protect, but they can also exacerbate infection and disease. So working with Rafi Ahmed, who had started a cohort of subjects with acute dengue infection in India, we performed this first study. And what we observed was that there were no correlations with disease severity that we could find, except for the abundance of the afucosylated glycoform on dengue reactive antibody here in red. So TH means thrombocytopenic. So that's a sign of very severe dengue disease. At both the early and convalescent time points, people with more severe disease had an enrichment for this afucosylated glycoform, also an enrichment for an activating subclass distribution. So just suggesting overall kind of the most activating FC domain repertoire that one could have. And when you look at how this correlated with disease, so increasing circle size here is lower platelet count, a sign of very severe dengue disease. So people with the highest level of the afucosylated form and the greatest activating subclass ratio had the lowest platelet count, and the dengue hemorrhagic fever 3 being the most severe clinical diagnosis, also the most severe clinical diagnosis. And we were able to show in a mouse model that these antibodies actually could result in platelet depletion. So a subsequent study that we performed was had the goal of isolating the antibody form in a human system. And the way that we did this was to look at maternal infant immunity. So because IgGs are transferred across the placenta, we could ask, does the maternal antibody repertoire correlate at all with disease phenotype in the infant? And the answer was yes. And we observed, so this is now maternal anti-dengue antibody stratified by whether their infants had symptoms during a primary infection. These are just control pooled IVIG, 2 different pharmaceutical formulations of IVIG. And again, we observed this increase in the afucosylated glycoform in the context of infants with more severe dengue infection. Now, as I mentioned, the afucosylated form has a very specific activity, which is increasing affinity for CD16, the activating Fc gamma receptor. So these data, which also have now been reproduced in multiple cohorts by different labs, they point to a role for CD16 in the pathogenesis of dengue infection. So to try and understand this role of CD16 in a controlled system, we needed some cell lines, because these cell lines, which are very commonly used to study dengue in the lab, don't express CD16. Though CD16 is expressed on host cells, primary cells that host dengue virus replication. So we made a panel of U937 cells, human monocytic cell lines, that express different combinations of CD32 and CD16, the 2 low affinity activating receptors, to ask the question, what is CD16 doing something that CD32 can't do? You know, why is there this clear association of more severe disease with a CD16 engaging antibody type? And then we performed a sort of classical experiment in dengue where you combine dengue with an antibody and you titrate the antibody, and there's this sort of curve that you can see where the infection is enhanced at some level. Here's what the infection looks like when the cells express neither activating receptor. When you express CD16 alone on the cells, there's actually no change, okay? So expression of that receptor on its own does nothing. Then you have CD32 alone, the other activating receptor. Well, there you do see a boost in infection. But when we combine the two receptors now, there's an even further increase in infectability of these cells. And here, just showing you 3 different monoclonal antibodies that bind to different regions of the dengue E protein. And here I'm just showing you one concentration of each monoclonal antibody, but using the 5 cell lines that we generated. Again, the highest level of infection in each cell line was when you express both CD32 and CD16. But if the CD16 does not signal, that is this white cell line. So same expression, but no signaling, it's functionally the same amount of infection as if there's no CD16 at all. So CD16 is required with CD32 for maximal infection, and signaling is required by that receptor. So overall, the model from this work became that afucosylation of dengue antibodies is a modulator of dengue disease severity, in particular with about a level of 10% being a cutoff for increased risk during infection. And at a molecular level, I did not show you all of the data that went into this work, but this is published. We found that CD32 is required for entry of the virus immune complex, which is why in the absence of CD32, we saw no increase in infection. But CD16, engaging CD16 and triggering that ITEM signaling exacerbates replication, promotes replication once the virus has entered. So that's an intrinsic enhancement of infection. Okay, so having clearly established, that this sort of role of afucosylation in dengue disease pathogenesis, we then wanted to understand whether there was a role for this glycoform or any glycoform in determining dengue vaccination efficacy. And so for this, we teamed up with our partners at Walter Reed and at University of Rhode Island, Alan Rothman and Nelson Michael, to study the response to Dengvaxia. And I'll just show you the study cohort here. So we have people at a pre-vaccination time point, so we'll call that baseline. They were then administered 3 doses of CYD-TDV, which is Dengvaxia, the Dengvaxia vaccine. And then we're comparing baseline and what we call T1 this is month 13, so 1 month post 3 doses of Dengvaxia. And what we did was to compare 2 groups of people. So this is obviously, you would need thousands of people to study disease severity. But what we could do with this cohort is just to study people who were protected or not from any infection after vaccination. So we studied people who became infected in the first year after dengue vaccination, the first year being a time that has been described as relative protection, versus people who remained uninfected for 6 years. So we thought that by comparing these 2 groups, we might see 2 extremes of immunity around dengue vaccination. So I'm just going to show you a few pieces of data from this work that was recently published in Science Translational Medicine. Comparing the FC domain structures, okay, at baseline pre-vaccination and at month 13, so 1 month post 3 doses, we do see some subtle changes in subclass distribution. This is of anti-dengue IgG, dengue envelope protein, IgG, some slight shift in abundance of G1 and G2. But looking at the glycoforms now, what's quite striking is that there's no consistency within the group in any form except for the abundance of fucose. So the level of afucosylation just goes up a very small amount from baseline to post-vaccination. And when we then compare to address the question of whether this is a change that's specific to the dengue reactive antibody, that's what this is. So this is total IgG from donors, or I'm sorry, what this is dengue specific IgG versus total IgG that's been depleted for dengue reactive antibody. And we see that the change is on dengue reactive antibody, though it's very, very small. So from pre to post vaccination, the blue to the pink, there is this slight increase in afucosylation. No other glycoform changes after vaccination. What was interesting here is then looking at the stratification of people who remained uninfected for 6 years versus those who became infected after vaccination. At baseline, pre-vaccination, there's already an association with what we're calling vaccine efficacy. So people who remain uninfected have a lower level of afucosylated antibody. At post-vaccination, same finding. So at that T1 time point, people who remain uninfected have a lower level of afucosylation. And in fact, if you look at the pre-vaccination abundance of afucosylation versus the post-vaccination level of neutralizing antibody, this is quite interesting. So the relative risk, so if you have an abundance of afucosylated antibody of greater than 9%, your relative risk for infection after vaccination 2.35, which is actually the same as if you have a low neutralizing titer post-vaccination. And these are interesting tests to compare, I think, because they're different in that the abundance of afucosylation is quite specific, right? So if you look at people above a level of about 9, many of those people go on to become infected. But it's not very sensitive at all. So there are a lot of people who become infected who are under that level. But it's just the opposite of low neutralizing titers. So low neutralizing titers captures essentially everyone who's going to be infected, but most people with low neutralizing titers do not become infected. So the nice thing I think about the abundance of afucosylation is you could actually target people prior to vaccination. You could identify people who might require an additional boost or some type of supplemental treatment. Now, to understand whether there's a functional role for these antibodies elicited by Dengvaxia based on their level of fucosylation, we did the following experiment. So these are mice that express human Fc receptors infected with a dengue 2 in the presence of, we have to use these monoclonal antibodies to kind of immunocompromise the mice so that the virus will replicate. And we gave pools of Dengvaxia elicited IgG with either low or high levels of afucosylation. So this is endogenously produced, just human IgG. The Dengvaxia pools were selected to have equal binding to the challenge strain, but no neutralizing activity. So this is an experiment looking at non-neutralizing binding anti-dengue antibody. And what we see here is that the pools with higher afucosylation, again, just varying by about 5 to 10%, elicit higher titers of virus in blood, spleen, liver, kidney, and that this is dependent on the presence of CD16. So these mice express all receptors except CD16, and that increase goes away. So that the Dengvaxia elicited IgG that is non-neutralizing and enriched for afucosylation triggers increased virus production, is the conclusion here. And then we asked, how does this affect the host response? So we did a very similar experiment, but now looked at the murine IgG production during infection. And as you might expect, when we challenge with either 100 PFU or 10 to the 6 PFU, the murine response when they received the high afucosylation pools is higher, okay? So this all came together in the following way for us, that we think the data support a model in which elevated afucosylation at baseline or post-vaccination ultimately just results. I didn't show you the cellular data. There's cellular data in the paper as well. It results in higher viral titer during infection. And what we think this may mean is that given equivalent exposure in an endemic population, people with elevated levels of afucosylated antibody are just more likely to have a productive infection, because those antibodies facilitate virus production. And the readout for that is seroconversion, which is how they detect positive cases in these cohorts. Okay, so I'm going to summarize with just two points. One, very broad, so post-translational modification of IgG antibodies, including silation and fucosylation, alter antibody structure and function. And manipulating these post-translational modifications is a promising strategy for optimizing vaccine design and improving immunity. Specific to dengue virus immunity, afucosylated IgG antibodies are enriched in people who progress to severe disease, and they're associated with risk of infection after dengue vaccination. And this is all the more severe disease and increased replication. What we've shown is a CD16 dependent phenomenon, also meaning you could potentially target by blocking the interaction between the antibody and that receptor. And in fact, this has been done recently by the Ravitch group. They developed a nanobody that interrupts afucosylated antibody interaction with CD16, and they show that it prevents severe dengue disease in a humanized mouse model. So it's very nice. This is starting to be built on this work. Just want to show you our team. The people who led the Dengvaxia study are Usama Ashraf and Shaborni Chakraborty, and our funding, and this is the rest of our wonderful group here. Thank you.
Again, I'm sorry I'm asking the first question, but it's really fascinating. Have you looked at the global fucosylation state of antibodies in chronic diseases like obesity, inflammatory diseases, aging, for example?
So aging, yes. Aging, yeah. Clear increase in afucosylated forms with age. We become more inflammatory. Yeah. The antibodies clearly do. Obesity we're doing now with Mark as part of the CCHI. It's like, I think that's a really important study. What was the other category?
Generally chronic inflammation, but that's interesting. And what do you know about the expression of CD16 and CD32 in myeloid precursor cells in the bone marrow? Could these antibodies be reprogramming the stem cells and then having a long-lasting effect?
No, we haven't looked at the bone marrow. That is a niche that we need to. Yeah, there's a lot to discover there. It's a great question.
This link between the post-translational modification and the inflammatory state, I think, is really interesting. Does that work the other way around too, where perhaps a different propensity for inflammation dictates how these antibodies are glycosylated? And could that be confounding some of the population level observations about who perhaps gets afflicted by viral disease?
We are investigating that, like, in a number of ways, actually, but I don't know the answer yet. But yeah, I suspect that may be the case.
Question?
I think for the talk, I was actually very curious about, like, are there any B cell intrinsic changes in like metabolism or something like that that are happening with age that could be driving those changes in fucosylation and so on?
Absolutely. I think it's a great idea. We have a very active area in the lab studying regulation of the glycosyl transferases that are responsible for but there are a number of different levels at which they can be regulated. And the sialic acid in particular, there's some controversy over whether it's all happening in the B cell or whether there's some extra B cell regulation as well. But I think yes is the answer.
Thank you.
Mhm.
In terms of vaccine design, how could we design vaccines either adjuvant-wise or antigen-wise that are directing the B cell response towards afucosylated or against afucosylation?
Yeah, so we're doing a very careful study to look at this now, but one observation we had during COVID is actually that mRNA vaccines elicit sort of the profile that you see in youth, the highly silated, highly fucosylated form. It was a little bit hard to, so we're doing a careful study to compare different vaccine platforms now, but there does seem to be a difference, and why that is, I don't know, but platforms do seem to make a difference.
In which way, or do you know? Can you mention the trend of what?
Well, we saw with
vaccine?
with Pfizer and Moderna SARS-CoV-2 vaccines in the early pandemic that they were enriched for both of those sugars, sialic acid and the fucose. Caveat there is I'm comparing to data from influenza vaccination, seasonal influenza vaccination, Fluzone. So different, different targets of the vaccines, but I think the data are comparable. And anti-HA versus anti-spike, but I think they are comparable. Anyway, we're doing it in a careful way now.
All right.
Thank you.
Thank you so much, Taia.